the mechanisms which are shown are the mechanisms which are shown are very difficult to remember, or whatever it is. But we have few tricks which we can implement in remembering these complement pathways, or in fact, the whole of immunology into a very simple, very interesting thing. Okay, let's start with our today's class, that is the complement system. Now, when I say complement system, these are, what do you mean is a complement system? These are a set of proteins which are present in the blood, along with the RBC, along with the WBC, that is, they are free flowing in the blood. So they get activated only when they encounter an antigen or an antigen antibody complex. Now you can get a doubt that when I say, madam, why are you saying it's an antigen and an antigen antibody complex? Well, the pathways in which the complement get activated is different. Okay, so there are different pathways by which the complement gets activated. Okay, that's why I said it can be an antigen or an antigen antibody complex. Okay, moving forward into the class, we'll go through the complement system in a different side headings. That is, the overview of today's topic will be, the first one we'll see about the history, that is the people who are responsible for identifying the complement and how did they use complement in the diagnostic purpose. Then we're going to see what are the complement components which are present, which are responsible for the destruction of the antigen, and which components or which pathways what we are going to see help in which type of immunity. That is it innate immunity or acquired immunity? What are the different pathways by which the complement gets activated? As I've told you in the previous slide, that is, there are different pathways by which complement gets activated. Then we'll see the net effect of an activated complement. How does this complement help in the destruction of the antigen? What are the different effects which brings about? And what are the effector functions of the complement? Now, is it mandatory for the complement to be regulated? That we'll see. What happens if there is a deficiency? We know vitamin D deficiency has a few symptoms, vitamin D has, etc. Similarly, complement deficiencies also can lead to some malfunctioning or disturbances in the immune status of an individual, right? Let's start with the history. Now, it was initially the person, that is Buchner, was the person who identified that, yes, there is a particular system or a particular set of proteins or a mechanism which is present in the serum, which is helping in the destruction of the microorganisms. Okay, he was the first person, that is, along with the other components. He said that there's something else which is present in the blood, which is helping in the destruction of the antigens which are entering into the body. Later, it was Jules Baudet. Now, he was the person who paved the way to identify that the particular protein what Buchner was talking about, that had both bacteriolysis property and hemolysis property. Said that this particular component which Buchner was talking had, or it uses both the antibody, which is a heat stable component, and a heat labile component. Now, what does heat labile mean? Heat labile means anything which can be destroyed when you heat it at 56 degrees centigrade for 30 minutes. Now, this is the standard which is followed for destruction of a complement. So when the serum was heated at 56 centigrade for 30 minutes, that particular component which was helping to destroy the antigen was not functioning. So he labeled it as heat labile. Okay, this component which Buchner was talking and which Baudet identified, it was actually given the name as elixir, but later it was Paul Ehrlich who coined the term complement. Later, because of the study which was done by Buchner, Baudet, and Paul Ehrlich, one more scientist called as Gingu. Okay, both of them, that is Baudet and Gingu, together in 1901, standardized a test which is used nowadays also as the complement fixation test, where the person's serum is detected for the presence of an antigen or an antibody in the presence of complement. Now, you'll be reading in detail about the complement fixation test when you go through the antigen antibody testing or the antigen antibody reactions. So what the people you have to remember are Buchner, who was the first and the foremost person or the pioneer for the identification of the components, complement components. Later it was Bordet who said that this component which Buchner was talking is heat labile. Okay. Later the term was coined by Paul Ehrlich, and the complement fixation test, that is the antigen antibody complex testing or the identification or the diagnostic test, was put forward by Bordet and Gengu together, right? So let's see what this complement is, what happens, or what are the main components or what main ingredients of a complement. This complement is actually made up of 30 different serum proteins. Now, all these 30 different serum proteins are put into 3 different categories. The first one is the components, that is the ones which are responsible for the complement pathways to take place. Then we have the propidine system. This is one more system which we'll be dealing in the later slides. Then we have the regulatory proteins. That is, these are the proteins which help to maintain the levels of the complement in a particular proportions in the serum so that they don't overact and destroy our own cells. Right? So the complement components are these 3 main, that is the complement components, the propidine system components, and the regulatory proteins. Let's see each one in detail. Okay? Let's see the first one, that is the complement components. What are these components? Actually, the different components of the complement are given as their number, that is numerically they're represented as C1 to C9. It goes in a sequence like C1, C2, C3, C4, C5, till C9. It is given those numbers based on the method or the process in which they are activated, in the sequence in which they are activated. We have only one component of the complement which gets or which has 3 more subunits, that is the C1 component. Okay. C1 component has 3 separate subunits, C1Q, C1R, and C1S. We'll see in detail how these components of the complement that is system help in the future, or how do they get activated individually. The next one is the propidine system and the regulatory proteins. Now, propidine system is nothing but the alternate pathway where the different factors, for example, factor B, I've given only one over here. We have factor B, factor P, factor D, which come into picture when the complement gets activated in this one more pathway, that is the alternate pathway. Then we have the regulatory proteins, as I've already told you, to regulate the proportions or the ratios of the complement in the serum. Okay. So these are the main things, what you need to remember. It is made up of 30 different serum proteins. Now, this 30 different serum proteins are grouped on 3 different categories. The first one is the complement components. Then we have the propidine system proteins, which help in the alternate pathway. Then we have the regulatory proteins, right? Okay, let's see the next one. Now, we've been reading about the complement components, etc. Now, we should know where this is synthesized, right? Now, we know that bone marrow is the main portion or the main area where the cells are synthesized. But where is complement synthesizing from? The major site for the synthesis of complement is the liver. Please do remember, the major site for the synthesis of the complement components or the proteins which are responsible for the complement activation, all those 30 different serum proteins are synthesized in the liver. Liver is the major site. Coming to the minor sites, now few amounts or few portions or the very little proportion of the complement can be got from the blood monocytes, macrophages, epithelial cells of the gastrointestinal tract and the genitourinary tract. Please mind you, this is very minor. The proportion is very minor. About 5 to 6% only gets from the minor sites. So whenever, if you have to answer the question like what is the major site for the synthesis of the complement components, your answer should be liver, not the rest, right? Okay. Now, we have been talking about the different pathways in which the complement gets activated, right? Now, complement system, we have talked about C1 to C9, they get activated in a different pathway or the sequence is different. Now, for the activation of these different components, the complement system has 3 different pathways: classical pathway, alternate pathway, and lectin pathway. Lectin pathway is otherwise called as the mannose binding lectin pathway. This is the latest pathway which has been identified in the latest times. So the classical pathway is also called as the classical cascade pathway, or just the cascade pathway. It is the age-old pathway. Then we have the alternate pathway, or the propidine pathway. Then we have the mannose binding lectin pathway, or the lectin pathway. Please do have this very clear in your mind. These three are different, okay? Three different pathways. They get activated differently, though the few steps are similar. They get activated into a different pathway, right? Now, let's see how a complement system gets activated, right? Anything should be triggered, right? For you people to study, there should be some trigger like exam, some assignments should be there. Similarly, there should be a trigger molecule or a trigger pathway for or a trigger point for these components to be activated. Now, what are these? That is, how does this go about? That is, once you trigger the activation, how it goes about. Now, it has major 4 different steps, as it is already, that is, you can see, right? Initiation of pathway, C3 convertase formation, C5 convertase formation, and MAC formation, that is membrane attack complex. This is the final step. Okay, so please be very clear with all the 4 steps. The first one is the initiation of pathway. Then we have the convertase formation. We have C5 convertase formation and the membrane attack formation, or the membrane attack complex formation. Please do remember, you have seen all these pathways, right? The classical pathway, the alternate pathway, the lectin pathway. Now, these pathways also have the same old, that is, these 4 steps only, but they differ in these 2 steps. That is, these 2 steps will be there, but the they get activated. That is, a different pathway will be there for the activation of the classical pathway. That is the initiation step, and the C3 convertase formation step will be different. Okay, I'll tell you what it is and how it goes about. Let's see the first one, that is the initiation of the classical pathway. Now, when I say classical, that means it is a pathway where the classical, that is the age-old process or age-old method takes place over there. Now, in the initiation of the classical pathway, you know very well that the antigen has to come into the body, right? When a person gets infected, the antigen is present in his body. Now, this antigen is counteracted by the circulating antibodies. The antibodies can be of different types: IgM, IgG, IgA, IgE, and IgD. Now, which is the first immunoglobulin which comes into participation? If I can get the answer. Which is the first immunoglobulin which comes into participation or for the rescue of the person? Well, it is immunoglobulin M which comes to the rescue. Then that is the first immunoglobulin which comes for the rescue. But that's why it can be seen in the acute phase of infection. But during the latent stages, that is during the, as the time process goes on, or as the infection proceeds, during the terminal stages of the infection, the next immunoglobulin which comes into picture is immunoglobulin G. That is the one which is seen in the last stages or the latent stages of the infection. So apart from these immunoglobulins, we have immunoglobulin A, immunoglobulin E, and immunoglobulin D. Okay, now all these 3, that is IgE, IgA, and IgD, they do not participate in the activation of the complement. Please do remember this very clearly. It is only immunoglobulin M and immunoglobulin G which participate in the complement activation, but not IgE, IgD, and IgA, right? Now, in the IgM and IgG, which one is quicker or which one is better? I'll say it is IgM. Okay. Now, if you can remember this picture of you, if you know the picture which is given over here, okay, the one which I'm showing or pointing out is the antigen. Okay, this antigen will have the complexes which are present on the surface. These are called as the, what are the antigen, recognizing points? Is it a paratope or an epitope? Okay, so these are the antigen specific molecules which are present on the surface of the antigen. Just think that this is a surface of a microorganism. So the surface of a microorganism is attacked by the immunoglobulin, that is the antibody. This is quite clear. The antibody comes and sits onto the antigen, right? Now we have an antigen antibody complex which is formed over here. Now once the antigen and an antibody complex is formed, the first component of the complement comes into picture. What is that first component? It is the C1. As the number goes, first component is the C1. So the C1 is a complex molecule. You can see it is a very complex molecule, right? The whole thing is C1. But what is C1? C1 is a complex molecule. You can see filamentous or globular structures over here, 1, 2, 3, 4, 5, 6. These are the 6 structures which you can see. So the C1 is what do you say? It is a complex molecule which is made up of C1Q, C1R, and S. Now, why did I label, or why is it given a bar over RNS? Is because of its enzymatic activity. Always remember, when a complement component or during the pathway, if there is a bar which is present, that means it is a enzymatic activity, or it is enzymatically active. Now, C1 complement or C1 component is made up of C1Q, C1R, and C1S. It is a complex. C1 is an hexamer, where you can see 1, 2, 3, 4, 5 spoke-like structures, and along with that we have C1R and C1S. The green ones, what you see is the C1R and C1S. Now, what happens over here? The first component is C1, which is coming into picture, right? The C1 is coming and sitting onto the FC terminal of the immunoglobulin. Now, if you know the structure of the immunoglobulin has 2 different subunits, FC subunit and FAB subunit. FAB subunit is the one which binds onto the antigenic component or the antigenic receptors which are present on the surface of the immunoglobulin. Then we have the FC component, which is free, right? Now, this FC component will have the receptors for the activation of the complement, or they get activated, or they can be shown only after the antigen combines with the antibody. So what happens initially? The antigen is present. We have the antibody. Antigen is combining with the antibody. But once this combination occurs, the receptor over here for the complement gets activated. These are called as the complement receptors, which are present in the FC terminal of the immunoglobulin. Now, this one, if it is clear, we'll proceed to the next one. I'll repeat it again. The first component which is coming into picture is C1. Okay. So when does that C1 come into picture, or when does it attach to the FAB terminal of an immunoglobulin? It is only after the antigen binds with the specific antibody. Okay. Now, once it binds with the specific antibody, near the FC terminal, we have certain receptors or certain domains which are present, which open up. That is, the shutter for them will be open. So once the shutter for the complement opens up, the first component, that is the C1Q, comes and it attaches to the components. Now you can see 6 spokes over here. So minimum of 2 spokes should be attached for a complement to get activated. So when we say 2 globules should be attached, if it is IgM or Ig, it is only immunoglobulin M. As we know the structure, it is 5. That is, it has 5 immunoglobulins attached to it. So it is always immunoglobulin M, which is better for the attachment of, or for the activation of complement. Then we have IgG, which is coming into picture, right? So after this one, the next step, same thing is forward over here. You can see the antigen. Okay, you have the receptors which are present, or the antigenic moieties, which can be identified by the immunoglobulin. Once the immunoglobulin comes and sits, it has an active receptor over here on the FC terminal of the immunoglobulin molecule, which gets activated. To that, we have the C1Q molecule, which is coming into picture, right? Now, once the C1Q attaches, we have other 2 small globules which are present, that is C1R and C1S. Now, these 2, that is C1R and C1S, are actually the esterase enzymes. Okay, these are the esterase enzymes which proceed forward. Okay. Now, one important point I want to tell you over here is C1 gets activated in the presence of calcium ion. It gets activated in the presence of calcium ion. Now, this is in just about how the activation process takes place. Okay, we'll see a small video after that, after this one, where it will be much better for you people to understand how it is, right? So let's move forward. You just follow how it goes on. Just follow the cursor. We have the antigen and the antibody complex which is formed. Now, once the antigen antibody complex is formed, the first molecule which comes into picture is C1q, right? The C1q is coming into picture. Now, once C1q is acted or it is attaching to the antigen antibody molecule, we have C1q, we have R and S. I've drawn a bar over here that indicates it is enzymatically active. So now we have the C1 which is active over here, right? Now, once the C1 is activated, the next one, that is this particular C1 molecule, it acts upon the next component, that is the C2. Now, this C2 molecule breaks open into C2A and C2B. Okay. Now, before this one, the C1 actually acts upon the C4 molecule. Okay. So, after the antigen antibody complex is formed, we have the C1 molecule which is activating. Now, once the C1 is activated, we have the R and S molecules which have the enzymatic activity. It acts upon the C4 molecule. Now, once the C4 molecule is activated or it is acted upon by the C1 complex, because this is enzymatically active, C1 breaks open into C4A and C4B. So we have the C4 molecule, which is breaking into C4A and C4B. Now, the C4B molecule is heavier. So it attaches to the complement. You can see, right? Over here, it is attaching to the complement cascade. C2A, C4A is lighter. Always remember, A is lighter, so it goes away. B is heavier, binding. B stands for binding, so it binds, right? C4B binds onto the complement pathway. Now, the next molecule which is coming over here is C2. Okay. Now, over here only there's a small disturbance about C1. Then it is not followed by C2, it is C4. So we have C1, C4, then we have C2 molecule which is coming into picture. Now, once the C2 molecule comes into picture, the C2 again is broken upon into C2A and C2B. Over here, C2A is heavier. So it attaches to the complement cascade. C2B is released into the circulation, right? So this particular molecule is called as a C3 convertase. This is the actual step which helps for the complement system to proceed further, right? So we have the C3 convertase over here. Now this C3 convertase, as the term suggests, C3 convertase. So it acts upon C3. So this particular molecule or the complex acts on C3, breaking down C3 into C3A and C3B. Now this C3B is much heavier. So it goes and attaches to the complex. Is it clear? C4B, 2A and 3B. This particular complex is called as the C5 convertase. Okay. Now, as the term suggests, it is C5 convertase. So it acts upon the C5 molecule. So till here, that is till the C5 convertase, it is one part of the classical complement pathway. Then we have the next aspect, that is the C5, which is coming into picture. Once the C5 is attacked by the C5 convertase complex, it again breaks open into C5B and C5A. C5B is heavier, it binds. So after C5B is broken down, now C5B is very, it is not stable, it is very unstable. So it requires for the stabilization. So the next component, C6, C7, C8, C9, they come into picture, they attach themselves onto the C5B, making it stabilized. Then we have the C9, which is the important component, which helps in making holes in the complement, in the antigen, which is called as a membrane attack complex. But this will be much clear for you if we see this one. Just a second, let me show the Is it visible? Akhil, can you see it?
No, ma'am.
Hello?
No,
you
ma'am.
cannot see it. Oh, just a minute.
For given that share sound and optimize for video clip, that option, what I told you earlier.
Yeah. I cannot see that now.
Ma'am, do one thing. Stop that share screen.
Okay.
Then again you do once more. Again you give share screen. There in down part you will see optimize video clip and
it's actually it's tick. That is it is ticked.
And now you try to share that video clip. Yeah. Now it's
visible.
Yeah, now it's visible. Yeah.
Activate the classical pathway.
Right.
While one
Now this is the complement pathway, which I want to, this is a small video which shows you, which gives you a clarity about how it is going to happen, right? Right. Immunoglobulin G, which is coming and sitting onto the, what do you say, the antigen. Okay, the green things, what you see over here, this is the antigen which is present. Then we have the immunoglobulin, which is coming into picture. Actually, in this video, it is showing as immunoglobulin G. Otherwise, it is immunoglobulin M, which is much better, right? So once we have the green one is an antigen, we have the immunoglobulin, which is coming into picture. We have an antigen antibody complex. Now, the next component which is coming into picture is C1, okay? Right, we can see the C1 component, which has the hexamer, which is coming and sitting onto the receptors, which is present on the FC terminal of the immunoglobulin. Now, once they sit, the next two, that is the C1s and C1r gets activated. Okay, once we have the C1s and C1r getting activated, they go and act on the next molecule. That is the next component of the complement. What is the next component? It is the C4 component, which is coming into picture. We see the C4 component coming into picture, right? Once the C4 component comes and sits, the C4 component is broken into two different moieties, that is C4b and C4a. C4a is lighter, it is going away. The C4b being heavier, it attaches itself onto the antigen, that is onto the surface of the antigen. The next one is C2, right? It again breaks open into C2a and C2b. C2a over here is heavier, so it is coming and sitting. It is attaching, or it is cozily sitting next to C2a, sorry, C4b, right? So we have these two components which are coming and sitting. Now, what is this one called? This one is called as the C3 convertase. That is C4b and C2a is called as the C3 convertase. Now we have the C3 molecule which is coming into picture. Now this C3 molecule is breaking up into C3a and C3b. C being heavier, it attaches to its friends, that is C4b, 2a, and C3b is attaching. This one is called as the, what is the next component which is coming? It is the C5 component. So this one is called as the C5 convertase. That is C4b, C2a, C3b. This is called as the C5 convertase, right? Now, the next component which is coming into picture, the one in pink, it is C5. Again, C5 comes, it sits, it breaks open into C5b and C5a. C5a is lighter. Now, we'll see what happens to all these lighter units in the later slides, okay? We'll see what happens to them later. We'll see this later. We'll go to the last step of the complement activation, right? So what happens once we have the C5 coming into picture? C5 is again sitting causally with its friends. The next components which are coming into picture are C6, C7, C8, and C9. I've told you C5b, it is very unstable. That is, it does not have stability. So it has to be attached to the other components, that is C6, C7, C8, and C9. So these are the components which are coming and attaching themselves to C5b. Remember, 6, 7, 8, and 9, they do not break open into C6, C7, C8. Now, once the C8 comes and attaches, it makes a small pore for the attachment of C9 molecules, right? C9 molecules are coming. You can see the C9 molecules which are coming and settling in the form of a hole. That is, they're making a bigger hole for the, entry of the water molecules or the ions, other ions into the antigens. So we have the pore which is formed, that is the small pore which is formed by the C8 molecule. Later, we have the C9 molecule which is coming into picture. It is making a bigger hole which allows the easy passage of or inlet of the water molecules. That is, there will be osmotic differences which is being happening outside and inside the the microorganisms. Because of this, we have we have the activation of the classical complement which is going to happen once the C9 gets attached to the classical pathway. We have the membrane attack complex which is formed, which you have seen in the form of a yellow colored structure which is making a big hole. That is called as the membrane attack complex. So once the hole is formed, there will be cytolysis which is going to happen, and there will be death of the microorganism. So that is the end result, that is killing of the microorganism happens. Right? The same thing which is put in a diagrammatic form, we have a C5 which is enzymatically active. It gets attached to C6, C7 and the C8 molecules, right? We have the C5b, 6, 7, 8 molecules which are coming and attaching. Then we have the poly C9 molecule, which is making a big hole into the microorganism or the cell wall of the microorganism. We have a big hole which is formed. So that leads to plasmolysis, that is the cytolysis of the cell, right? That is about the classical pathway. As the term suggests, it is classical. That means it involves the antigen along with the antibody. It is taking the antibody along with it for the activation of the classical pathway of the complement. That is, it starts from C1, it ends at 9, right? That is the classical pathway where all the components get involved. The next one is the alternate pathway. This one is a bit different than the classical pathway. In what steps is it different? As I've already told you previously, only the first 2 steps, that is the initiation of the complement pathway and the C5 convertase formation only, it differs. Let's see what happens. Now, these are few things which you can remember in your bullet points. That is, it is antibody free. Okay, always remember the alternate pathway is free of the antibodies. That is, it does not require the antibody participation for the activation. It does not require the participation of antibody for the activation of the classical pathway. Now, this takes place in 4 steps. Okay. Actually, it differs in the first 2 steps for the classical pathway. That is, the C1, C2, and the C4 are not involved over here. When we have seen the classical pathway, where C1, C2, C3, C4, they're coming in sequence, right? C1, C4, C2 comes in sequence. This does not happen in the alternate pathway. Apart from that, there are 3 main factors which are present instead of C1, C2, C4. They are factor B, factor D, propidine. These are the factors which mainly help in the stabilization of the C3 molecule, and the further activation or the further proceedings of the complement pathway activation by the alternate method takes place by these 3 components. Always remember, C1, C2, C4 don't play a role in alternate pathway. What plays a role is factor B, factor D, and propidine. Now you can ask me a question, madam, if you say the antigen antibody complex is not there, that is, we don't require the participation of an antibody over here, then how does the complement get activated? You have told us once the antibody comes and sits, the gate for the complement attachment of C1 takes place. That is what happens in classical pathway. But in alternate pathway, the antigens which are responsible for the activation of the alternate pathway are different. Okay, what are the antigens which help in the participation or in the activation of the classical pathway? The antigens which are mainly responsible for the activation of the alternate pathway are LPS. LPS stands for the lipopolysaccharide, which is an integral part of the gram-negative cell wall. Then we have teichoic acid, which is an integral part of the gram-positive cell wall. We have yeast cells and we have infected viral cells. All these are the ones which are mainly responsible for the activation of alternate pathway. Along with this, the endotoxin. Now, in all these four, which one acts as an endotoxin? If you can give me an answer, it is lipopolysaccharide which acts as an endotoxin, because gram-negative organisms are the ones which have LPS or the lipopolysaccharide. Okay, that one acts as an endotoxin, right? So because it is not released into the external environment, it acts as an endotoxin. Now, once these antigens, they are present